Hello, welcome to 13 Universal, and this is Shedrack. Shedro, that's the right name to call me, Shedro. So, we are starting with cell structure part one. We are going into content specific um, classes and all of that. So, get ready. So, this is cell structure. We are going to be solving this question. I'm going to be doing some questions with it. So we treat this topic and we solve questions, knowledge check. Okay. So the first question I'm going to ask you is, what is a cell? So what is a cell? What is cell structure? Well, a cell, like it is said, a cell is a basic or fundamental structural functional and functional unit of life. That's it. So the next question I'm going to ask you is, what are cell organelles? If I look at it, now, these are cells. Okay, you can see these cells. So what are cell organelles? You can see that these cells have some structures inside of it. So cell organelles are they are um, part of cells, all right, tiny structures found inside a cell that have unique function and structure. Okay, so if I ask you again, what types of cells are these? Okay, now these cells, of course, you can look at this. This is plant cell, has cell wall. This animal cell, this is bacterial cell. So these two cells are eukaryotic cells, okay, because they have nucleus. They contain genetic material inside nucleus, just like they all have nuclear membrane. Okay, all right, this doesn't have nuclear membrane, it has cytoplasmic DNA. All right, let's go. So in this broadcast, in this episode, we're going to be looking at microscopy. We're starting with microscopy. So what is microscopy? Okay, so it is simply defined as the use of microscope to view, observe, measure, and draw specimens, right? Cell structures are very tiny and microscopic. So in this episode, our objective is that we're going to be discussing a lot of things about how to use microscope, talking about magnification, resolution, cell fractionation, and plant diagram. All right, so let's go. So this is an example of a microscope. Okay, so my question is, what type of microscope is this? All right, look at it. What type of microscope is this? Of course, this is light microscope. Okay, look at the part. This is the stage where you put slide the specimen. These are lenses. So these lenses are called objective lenses. Okay, so they have different magnification of times 4, times 100, times 40. So this is actually another lens. This lens here, these lenses are called eyepiece lenses. Okay, so they also have their own magnification times 4. All right, no, times 10, and maybe times 5 too. Okay, so you just put your stage, your slide here, contain the specimen, and then you start focusing with coarse adjustment knob. And once you find the image, you make it, very sharp using fine adjustment. Okay, all right. So now remember, every microscope has three two major property, right? Whether it is light microscope or electron, it has magnification. Okay, I'll call it M, and it has resolution, I'll call it R. Okay, so I'm going to talk about magnification here. I'm just going to open it up. So what is magnification? Magnification simply means what? Answer the question. Okay, the number of times an image is bigger or larger than the specimen, the number of times. Okay, so if you check here, all of these lenses, watch this, these lenses, they magnify, all right, they make the specimen bigger, the image is bigger. All right, so like I told you, we have if you look at what I've told you here. All right, guys, so of course this is times objective, we'll call it the eyepiece objective, sorry, the eyepiece lens, these objective lenses. So if you look at this, for example, we can have times, all right, 10 here, right, or times 4, any one of them. So that is the magnification. Let's say times 5 here. Okay, so that could be there. If you look at this could be times 10. That means it magnifies object by 10 times of its original size. This could be times 40. Now times 40 here is called high power. Times 10 is called low power. Okay, we could have times 4, which is called scanning power. All right, so whatever you use to view, so see guys, watch this. Now this is objective, these are objective lenses. They have their own different magnification. So whatever you use, for example, if you use times 10 to view a specimen on a slide here, let me show you, because you have a slide here, all right, placed on the microscope, is under the light. Maybe this is your specimen, and then you now position the objective lens. So whatever the objective lens sees, it magnifies it. For example, if you're using times 10 objective lens, times 10 magnification, okay, it will increase it by 10 times its actual size. Then if you're using times 10, maybe this is times 10, times 10 will further increase the size of the image 10 times. Okay, all right, let's assume that's times 10. This is times 5. Okay, so you can see, guys, you can see, let's assume we're using this and this. So whatever this is this magnifies, of course, magnifies it by 10 times. So because this also has it has lens and magnifying power, so this will magnify 10 times of the image. Okay, so this has already done 10 times. This will magnify that 10 times by 10 times. So the total magnification will now be 10 times 10, that's times 100. Okay, so if I'm using this and this objective lens times 10 objective lens and times 5 objective lens, so the total magnification will be 10 times, all right, 5. All right, if I decide to use times 40 and objective lens and times 10 eyepiece, so I multiply times 10 eyepiece with times 40 objective lens. So guys, what I'm trying to say here typically is that of course, magnification, now these are facts about magnification, all right, is directly proportional to size. So that means once magnification increases, once I move this, for example, if this is times 10, once I increase it from times 10 to like times 40, the size will increase. Of course, this is times. The size of the cell increases about 4 times bigger. So the factor here is 4. So it increases 4 times bigger. Is that okay? So that's a factor. All right. So that's what we're trying to say, that once of course magnification increases, the size will increase. So the highest magnification will give us the biggest size. However, there's another fact about magnification. The magnification, all right, magnification is indirectly proportional to number of cells. So the bigger the magnification, the less number, all right, the cells you see. Okay, so for example, if at times 10 I could see with I could see like let's say 100 cells. Okay, now let me say, okay, 40 cells. Yeah, 40 cells at times 10. When I increase it to times 40, the number of cells will decrease by 4. Okay, that is divided by 4. So I can only see 40 divided by 4 will give me like 4 cells. Okay, so it's going to decrease. All right, so because if I increase it by 4 from 10 to 40, right, and this number of cells, if I increase this to this, the number of cells, whatever I have here, all right, divided by 4. Okay, 40 divided by 4 is not 4. 4 divided by 4 is 10. Yeah, so the number of cells will now decrease to 10. Okay, all right. So when you increase the magnification, the number of cells decreases. All right, let's go to resolution. Of course, this is resolution. Now, what is resolution, guys? Resolution is what? The ability of a microscope to distinguish between two separate points. So the ability of microscope to tell us that these 2 point A, right, and to point B, right, they are not the same. That is resolution. Now I want to say this, that resolution deals with details. Okay, I forgot to tell you that magnification deals with size, so resolution deals with details. Anytime you're dealing with a question asking you what you can see and what you cannot see, all right, that has to do with resolution. Okay, anytime you're dealing with a question asking you size, all right, okay, that's magnification. Now guys, I want to say this very importantly, that resolution is not affected by magnification. So if I call it R, so there's no relationship. It's not affected by magnification, not affected. There's no relationship between them at all. No relationship. No relationship between them. That means that even if you increase magnification, it does not affect resolution. Okay? Now, resolution, guys, you can see, very important, is a product of wavelength. So resolution is indirectly proportional to the wavelength of radiation. Every microscope has radiation used to see. Okay, now our eyes can see, our eyes can only use visible light to see. Okay, now light microscope can also see using visible light. All right, visible light has a range of course, these are range of visible light. The smallest range is 400 nanometers. This is the wavelength. Okay, the highest is 700, that's red, and all of that is violet. Now of course, these are different electromagnetic waves that could be used to see, but they have their different wavelength. So gamma ray, x-ray, UV, you see that resolution is indirectly proportional to wavelength. That means the shorter the wavelength, the higher the resolution. Okay, so gamma ray, for example, has short wavelength 0.1 nanometer. It has higher resolution than UV ray, which is 10 nanometers. It can see more, okay, because it can see, all right, objects that are less than it. Okay, you can, sorry, objects that are bigger than 0.1 nanometers and above. Okay, for example, it can see 8 nanometers, it can see 6 nanometers, it can see 1 nanometer, but 10 nanometers cannot see object less than this size. Okay, you can see only those above 10 nanometers. So this sees more, it has high resolution. All right, but you see two of them have high resolution than visible light, because you see the wavelength is 400. That's a lot. Okay, the higher the wavelength, the lower the resolution. Okay, so electron is between 0.1 nanometers to about 0.005 nanometer, kind of. Okay, so it has, of course, I mean the resolution is 0.1 nanometer to about 0.5, so you can see a lot. All right, let's see. So you see that resolution is in the proportional to wavelength. Okay, very, very important. Now another thing I want you to learn is that resolution is a product of wavelength. We mentioned that. Okay, now guys, like I told you, let me mention it again. Now resolution, all right, if a microscope has a resolution that is smaller, if the resolution is smaller than the size of the object, okay, all right, what that means is that the object can be seen. It can be seen using that microscope. Once the resolution, of course, the resolution of electron microscope, right, like I've mentioned, the resolution is about what mentioned for me, 0.5 nanometers. That's resolution. That's not wavelength but resolution. Okay, wavelength is converted to resolution by calculation. All right, the resolution of light microscope is about 200 nanometers, right? So you can see based on this, right, 200 nanometers. So the one that has of course, this size is not, it doesn't determine detail. What determines detail, of course, the smaller the resolution, of course, the more I mean the value of resolution. Okay, for example, if light microscope is resolution is 200 nanometers and they're trying to use it to view an object, right, whose size, now this nanometers, if the object size is about, okay, give me one, like, okay, 100 nanometers, right, can you see that that is not visible, right? So what I'm trying to say is that if the resolution, watch this, let me take it again, if the resolution is greater than the size of the structure, what it means is that it cannot be seen, not seen. You cannot see the object using that particular microscope. All right, not seen. See what I'm trying to say here, guys? Hmm? See, this is a law. If resolution of a microscope is less than the size of the structure, then you can see the structure. As long as the resolution is less than it, okay? But if the resolution is greater than it, you cannot see it. So for example here, the resolution of light microscope is 200. This is 200 nanometers. That of structure, okay, is 100 nanometers. This is greater than this, so this light microscope cannot resolve this. All right, come here. Now this is light microscope, okay? This is the wavelength of light. The smallest wavelength is 14 nanometers. Wavelength is the distance between two peaks. Is that okay? Yeah. Okay, in a wave, right? So you see that wavelength is smaller than the structure. The structure of this particular, the size of this structure, okay, is 25 nanometers. So remember, if it is less than the wavelength or less than the resolution, it cannot be seen. So because this structure here is 25 nanometers, wavelength is 200 millimeters. Of course, resolution of light microscope is 200 nanometers. Wavelength is 400 nanometers. Resolution is 200 nanometers. You see that the resolution is also bigger. As long as the resolution is bigger than the size, it cannot be seen. So light microscope cannot be used to see this structure whose size is 25 nanometers. Okay, why? Because once the resolution is greater than the size, it cannot be seen. The same thing, once the wavelength is greater than the size, you cannot see. Why? Because the wave will not be able to interfere. Look at, see, stained ribosome of diameter 25 nanometers do not interfere with light waves. Why doesn't it interfere with light waves? Because it's too small. All right, so it cannot interfere because the size of this, all right, ribosome is smaller than the wavelength and even the resolution. So light microscope cannot see 25 nanometer ribosome. But look at this. This is stained, see it? Stained mitochondrion of diameter 1000 nanometer interface with light waves. Can you see that the size of the stained mitochondria is 1000, so is greater than even the wavelength 400 nanometers. And remember what I told you is a law. Once resolution is less than, okay, the size, you can see it. Resolution is 200 nanometers. The size is what? 1000. So once the resolution is less than that, you can see it. Okay, so this can be seen. Why? Because the mitochondrion here will interfere with the wavelength, with the light waves. Okay, once the size of structure is bigger than the wavelength, bigger than the resolution, then it can be seen because they can interfere. But once it's less than, it cannot be seen. Okay, so let's move to the next slide quickly. Knowledge check one. Okay, this is a knowledge check. Just knowledge check. Which combination of lenses for light microscope will give the greatest magnification? So which one gives us greatest magnification? Remember, guys, that pause the video and try it. Okay, tell me your answer. All right, this is eyepiece lens. Okay, so whatever is an eyepiece lens multiplied by whatever you are viewing with objective lens. Objective lens is the one that views the specimen on the stage of microscope. Whatever it is viewed is further multiplied by what is on eyepiece. So the total magnification will be the magnification on eyepiece lens times the magnification on objective lens. Okay, so quickly, without wasting time, so the total magnification of this option is 5 times 100, which is 500, right? Okay, yeah. For the second one, 10 times 40, which is 400, right? Okay, the third one, 15 times 40 will be what? Okay, that will be of course, that will be 600, right? Yeah, so 15 times 100, that will be 1500. So which one will give us the greatest magnification, guys? Right, so this is times 1500 times 600 times 400. Which one gives us the greatest magnification, guys? All right, of course is this, the 1500 will give us the greatest magnification. It will give us the greatest magnification, guys. All right. Now, if the question had come another way and tells us which of these magnification, all right, which combinations of lenses for a light microscope will give the greatest number of cells. Remember, the greatest number of cells. Okay, the higher the magnification, the lower the number of cells. Okay, that means the greatest number of cells will be given by the magnification that gives us the lowest the combination that gives us the lowest magnification. Which one is the lowest here? Of course, this. If they ask for the highest number of cells, but the question is talking about greatest magnification. Okay, the answer is delta. All right, this is knowledge check 2. What is defined? What is the definition of resolution of a light microscope? Okay, so pause the video and try it. Now, A says the degree of sharpness produced by microscope. Nope. The greatest distance between two objects visible in the same field of view. Well, nope. The minimum distance that allows two objects to be viewed as separate. Okay, makes sense. Yes. So the size of the smallest object visible using the microscope. Now, the magnification is the distance, all right, that allows two objects to view as separate. That is the ability to see to distinguish between two separate objects, right? So the answer is C. All right, knowledge check 3. Watch this, guys. A specimen is observed twice with a microscope. Firstly, using green light with wavelength of the wavelength is 510 nanometer, and then using red light with a wavelength of 650 nanometers. What happens to the magnification resolution when using red light compared to green light? Okay, so I want you to pause the video and try to answer this question. Pause this video, guys. Now, let me give you clues and you try. Watch this, guys. Let's play. Remember that resolution, okay, is indirectly proportional to what? Wavelength. Okay, so the higher the wavelength, the lower the resolution. If you check these two, this and this, which one is higher? Of course, this is higher. All right, so okay, because it has lower wavelength. Okay, longer wavelength, shorter wavelength has higher resolution than longer wavelength. So when you moved it from 510 nanometer to 650, the resolution decreased. So this is decrease in resolution. Okay, decrease. You are decreasing the resolution. All right, which option says decrease? Okay, this is decrease. So the one that says decrease is A. Yeah. No, for resolution. The one that says decrease for resolution is C. Yes. Okay. Now, resolution does not affect magnification. Magnification does not affect resolution. Okay. So resolution decreases and magnification remains the same. Okay. Are you there? So once you increase this length, the size of wavelength, resolution decreases. All right, and it has nothing to do with magnification. All right, look at this. What is the typical resolution of a microscope using daylight as a light source with a ×10 eyepiece lens and a ×40 eye objective lens? So what is the typical resolution? Remember, pause this video and check. Pause this video and check and solve it. Okay, remember that when you increase the magnification from ×10 IP, you see this has what is the typical resolution of microscope using daylight as a light source with ×10 IP, this ×10 eyepiece lens, and that's ×40 objective lens. Okay, the resolution does not change. Okay, resolution of light microscope is 200 nanometers, doesn't change. Okay, even if you increase the objective length, it doesn't change. It's not affected by magnification. Resolution is not affected by magnification. The same thing, magnification is not affected by resolution. So the resolution of light microscope is 200 nanometers, right? So knowledge check 2. knowledge check, another one. I think it's about the fourth one. So I want you to pause the video and solve this. So which feature is visible with a light microscope using natural light source? Okay, which one is visible? So pause the video. Okay, try this out, guys. Remember, guys, that resolution of light microscope, okay, of light microscope is always 200 nanometers. Okay, and remember what we said, guys. We're looking at what can be seen. That if resolution is less than, okay, size, then if it's less than, this is less than size, okay, less than size, then it can be seen. The object can be seen. But if it is greater, right, if it's greater than the size of the object, cannot be seen, right? So now if you check the first question, the first option here, the question says which feature is visible with a light, which one can be seen? Resolution of natural light source is 200. So if I bring 200 nanometers here, 200 nanometers is greater than that 2 nanometers of DNA molecule. Okay, so according to our law, if resolution is greater, right, than the size, then you cannot see it. It's not visible. This is not visible. If resolution, the size, the value of resolution is greater than the size of the specimen, you can't see it. Okay, now if you look at parameter is 200 micrometers, guys. I forgot to teach you that anytime you're moving from centimeter, because we have different units we use in microscope. If you're moving from centimeter to micrometers, guys, you know what? Okay, to millimeters. Let's say millimeters first. Yeah, okay, you multiply by 10. Okay, whatever you have it in centimeter times 10. If you're moving backward, you divide by 10. So if you're moving from millimeters to micrometer, just like U symbol, micrometers, you multiply by 1000. Okay, right? If you're going backwards, you multiply also, you divide. Anytime you're going backward, you divide by 1000. So if you're moving from micrometers to nanometers, you multiply by 1000 too. Okay, if you are going backwards, you divide. So look at this. So that's all right. All right, 1000. Okay, so if you want to convert this to nanometer, that's 200. Now 200 micrometers times 1000, that will give us 200,000 nanometers. That's very huge, guys. 200,000 nanometers. Okay, remember, 200,000 nanometers and 200 nanometers, which one is bigger? Right? Okay, so you see, watch this. So remember, if resolution is greater than the size, it cannot be seen, cannot see. If resolution is less than the size of structure, parametric cell diameter is 200 micrometers, which is same as 200,000 nanometers. 200,000 nanometers is bigger than 200 nanometer resolution, right? So you can see it. There can be interference with the wave, with the light waves here. But if you see phospholipid bilayer, which is 8 nanometer, is less than the resolution, so it cannot see it because the light waves will not interfere with phospholipid bilayer. So ribosome is 200 nanometers. Light microscope resolution is greater than the size, so you cannot see it. As long as resolution is greater than the size, you cannot see it. As long as the size is bigger than resolution, you can see it. That's the law. Let's go to the next question. Now, guys, you know we have types of microscope. We have light and electron. These are the differences between them. So you can see source of radiation here is light, is electron. So C with light here, C with electron here. Wavelength is 400 to 700. This wavelength is too short, man, very short. So this has small wavelength. All right, maximum resolution is 200 nanometers, and this is from plus or minus 0.1 to 0.5 nanometers. So this electron microscope has higher resolution. It can see a lot. It can see molecules from maybe like 1 nanometers and above. Okay, so watch. That's what we see. This can see. Remember, resolution has to be less than the um, the size of the structure for the structure to be seen. So this can see from minimum, of course, 1 nanometers, right, and above. Okay, 1 nanometers and above, or 0.5 nanometer and above. So you can see why this can see maybe from maybe 210 or 200 nanometers and above, less than 200 nanometers, right, and above. So and above. So you see that light microscope cannot see anything below 200 nanometers. Okay, but electron can see things way far below, way far below 200 nanometers. This cannot see it. Or you can see their lenses, glass, electromagnet. Now you see the advantage of electron microscope is that it has high resolution. You can see a lot. While the advantage of light microscope is that it has low resolution. It has not advantage. The advantage is that it can be used to see live specimen. Okay, live specimen. So you can use it to watch, like for example, mitosis. You can use it to watch live processes in real time. All right, electrons can only be used to view dead specimens, because electrons do not travel, okay, through light. It doesn't travel through live specimen. Why? Electrons can only travel through vacuum. Live specimen has water and air, so water and air scatters electron beams. So if you remove water and air, it becomes dead. So now see the type of stain: colored for light, heavy metal for electron, image, colored photomicrograph. This is black or white. Okay, so there's a lot. All right, let's go, guys. All right, now watch this. Like I've mentioned, I've just talked about this. Electron can travel only through vacuum. Okay, now magnification. Okay, this is the formula of magnification, guys. Let's go. So don't forget, magnification is always, like we say. Now if you want to calculate magnification, let me show you here. Okay, magnification, if magnification, it will be image over actual. If you're calculating actual, it will be image over magnification. If you're calculating actual, it will be actual times magnification. Okay, what that means is that you're going to have something like this. Okay, we have something like this. Yeah, so you see magnification. If I have an object like this, okay, all right, and I'm asked to calculate magnification, right, of this point, from point, maybe point X to Y. Guys, don't forget this. All right, so I was asked to do the magnification. The actual size of this is like maybe like 0.2, okay, micrometers. So the question is, I should calculate magnification. What is magnification? Okay, so that's the question. So what do you do first? What you do first is that you go to the image. This is the image. Use your ruler, measure from here to here. All right, whatever you have measured, convert it to micrometers. For example, if you measured it in millimeters, to move from millimeters to micrometers, you multiply by 1000. So measure first in millimeters, multiply by 1000 to convert, right, to micrometers. And then what you then do is that becomes your image size, and then divide it by the actual. Okay, divided by the actual. For example, if for example xy is 2 centimeters, right, that is the image size. Convert it. You convert it by multiplying with 10, of course, 1000. If you multiply by 1000, what are you going to have? Okay, so I want you to tell me, if you multiply by 1000, what will you have? So 2 centimeters times 1000 gives you what, guys? What does it give you? Of course, it's going to give you that's 2000 micrometers. So 2000 micrometers divided by, that's image size, by the actual. The actual is 0.2. All right, remember you must convert to micrometers. Okay, divide by 0.2, and then you will have what? So if you say 2000 divided by 0.2, get your calculator. 2000 divided by 0.2 gives you what? 10,000. So magnification here, right, which is, of course, you have to write the formula, is, image size over actual. Okay, so that's what we have done here. So the answer is times 10,000 times 10,000. Okay, that's magnification. Now that's if you have this. But if you have another object, guys, watch this, the same object, and then now give you a scale bar. Scale bar is a line with 2 bars. Okay, and then now put something here, maybe 2 micrometers, and they ask you, if you're dealing with scale bar, guys, the formula changes. Okay, even though they are similar at the end of the day. So if you want to calculate magnification of a scale bar, so the magnification is what you measure now changes. All right, so if you want to measure this, you measure the scale bar. So it's going to be length of scale bar, the length of the scale bar all over the value on scale bar. So that means you don't measure this, because scale bar has been given to you. If scale bar was not given to you, measure it, right? But here, measure the scale bar, length of the scale bar, convert it to micrometers, right, and then divide by the value on scale bar. So the length of scale bar is serving as image size. Whatever you measure with a ruler, measure the millimeters, multiply by 1000 to convert to micrometers, divided by the value written on it, which is 2 micrometers. Okay, so let's check out something here. So knowledge check. Let's see whether you are following. The actual length of a cell structure is 8 micrometers. 8 micrometers. Which steps are used to calculate the magnification of an electron micrograph of this cell structure? Okay, so I want you to pause the video, try this, and then let's see what you got. All right, pause the video and try it. So what do you got, guys? Now, of course, step 1 says measure the length of the cell structure image. Yeah, you measure it on in centimeter. Okay, okay, that's not bad, but let's see whether there's something that fits in there. All right, but normally measure in millimeters, but you can measure centimeters if that's what you have. Measure 2 says measure the length of the cell structure image on micrograph in millimeters. That's also correct. Now step 3 said divide the image, all right, length by 1000. You don't divide. So if you measure in millimeters, how do you convert millimeters to micrometers? Micrometers is the measure unit, so you multiply by 1000, you don't divide. But if you measure in centimeter, all right, if you want to convert centimeter to micrometers, you multiply by 10,000 from centimeter to micrometers. Okay, all right. But if you are working with millimeters, you want to convert millimeters to micrometers, you multiply by 1000. So there's no 10,000 here, therefore we cancel out this. Step 1 is out. So we go with step 2. After step 2, you multiply. So it's going to be 2, 4 multiplied by 1000, and then you divide. Okay, because that's your actual. Once you get your image, you divide the image by the actual. You don't multiply, you divide it. So we're going to have, of course, step 2. What disqualifies step 1 is because there's no place where they say we should multiply by 10,000. If you're converting centimeter to micrometer, you have to multiply by 10,000. All right, but now millimeters multiplied by 1000, so that's correct, and then divide by 8. So that's 145. 1 multiply, no, you don't divide by, sorry, this is wrong, you multiply by 8. Yes, so 146. Okay, 146 is B's answer. All right, so check this again. Now I want you to pause the video and try. The actual diameter of prokaryote is 0.5 micrometers. An actual electron micrograph of the cell has magnification of this. So what is the diameter of the cell in the image? Pause the video. They're asking us image size. What is the image size? So we're looking for image here. Okay, pause the video and try it. All right, if the magnification is image over actual, and we're looking for the actual now, sorry, for the image will be what? Will be magnification. That's the formula. Magnification times actual. Okay, so what is our magnification? Magnification is 50,000, right? That's it. So 50,000 times 50,000, right, times 50,000, right, yeah, multiply by 0.5. So 0.5 will give us multiply by, that's like half of it, which is 25, right? 25,000, right? Yeah. So convert it to what? All right, 50,000 divided by that. If converted to standard form, that is going to count 1, 2, 3, 4. Okay, now the answers are in millimeters. So what we have here is we have 25,000 in, okay, micrometers. So how do you convert micrometers to millimeters? You divide by 1,000. So 50,000 divided by 1,000, sorry, 25,000. We are moving from micrometers to, right, to millimeters. So you divide by 1,000, so that will give you 25 millimeters, right? So 25 millimeters, if you move the point from here to here to standard form, it's going to be 2.5 times 10 raised to the power 1, okay, which is C. All right, so check out this, don't waste time here. So see, same thing, the photomicrograph shows cells from a human blood smear. You can see, so which calculation shows a correct method to calculate the actual diameter of the neutrophil shown? So what do you mean? What do you do? Measure P and Q, right? So A says length of line PQ in millimeters, and then multiply by 1,000, and then divide by magnification. That's how you get the actual, because actual is image over magnification. Okay, all right, so that's what you do. Now B is wrong because millimeters should be multiplied by 1,000 to convert to micrometers. What is wrong with C? Magnification, all right, should not be divided. Of course, actual, remember that magnification, right, is equal to what? Image over actual. Is that okay? So that means actual, make actual sort of the formula, image over magnification. So that's what this disqualified C. What is disqualified C and D? Okay, that's what disqualified them. What is disqualified B is because you don't multiply it by 10,000 by 1,000. All right, so the answer is actually sorry, A is the answer. Okay, so let's go to, okay, look, see, we have a lot of knowledge check. A scale bar on electron micrograph is 2 cm long and represents an actual length of, okay, 1 micrometers. What is magnification of the electron micrograph? So check, now we have scale bar. Try to solve it, guys. From here to here, let's assume this is 2 centimeters, I guess. Okay, 2 centimeters. Okay, you measure it, and then we have the actual is 1 micrometers. Okay, so remember, magnification is length of scale bar, which they gave us, which is 2 centimeters. So convert 2 centimeters to micrometers. How do you convert 2 centimeters to micrometers? Like I taught you, multiply by 10,000. So that means this is going to be, okay, 20,000, right? Yeah, 20,000 micrometers, right, divided by 1 micrometer. That will give you times 20,000. The answer is C. Okay, all right, look at it again, don't waste time. So you shouldn't miss this. Close your eyes. The student was asked to use the scale bar shown to calculate the magnification of a cell on a photomicrograph. Which method could the student use to calculate magnification of the cell? So what do? You have to measure the scale bar, convert it to micrometers. Okay. Now look at A. Say divide the diameter of the cell by the length of the scale bar. Both measure the same unit. Nope. Okay. So measure the diameter of the cell in millimeters, multiply by 20,000, 2,000, and divide by, nope. So what do? Measure the length of the scale bar in millimeters. Just put your ruler here, measure from here to in millimeters, and then convert to micrometers. Yeah. How do you convert to micrometers? Multiply by 1,000 and then divide by 2. So once you convert to micrometers, that gives you length of scale bar. Then divide it by value of scale bar, which is 2. Right? Length of scale bar, measure the length of scale bar, convert to micrometers, divided by the value that gives it. Now, why is it wrong? Measure the length of the scale bar, convert to micrometers, multiply. No, you don't multiply by 2, you divide by 2. Okay. All right, let's go to something else. Now, we're looking at right now calibration of eyepiece graticle. Calibration of eyepiece graticle. Calibration of eyepiece graticle. Calibration of eyepiece graticle. So, guys, remember, if you want to measure something under the microscope, you need 2 scales. See this tiny one here? It's called eyepiece graticle. You see this big guy here, stage micrometer. So the eyepiece graticle is used for measurement. Okay, is usually embedded on. Remember, the eyepiece graticle is embedded on eyepiece lens. Okay, that's why it's called eyepiece graticle. It's on eyepiece lens. The stage micrometer is usually on a slide, and they put it on the stage of micrometer. Okay, now you use this to measure. Okay, and after measuring, you write out the number of eyepiece graticle. For example, if you check in here, from here to this is, all right, 20. From 0 to 1 is 10. 1 to 2 is, okay. Now if you check your stage micrometer, you can see that the same thing. Okay, so remember, you put this on the eyepiece graticle, on the eyepiece. This is put on eyepiece lens, and stage micrometer is on the stage. Okay, so you now clash 2 of them and find point of overlap. For example, point of overlap here, right, is here and here and even here. This is where the 2 division clashed. So 1 and 50 here, 60, and of course, if you check this from here, now eyepiece divisions from here to here is 10, 20, 30, 40, 45. Okay, so 45 eyepiece graticle divisions equals how many stage micrometer division? From here to here is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 45. So you have to clash them. All right, and then eyepiece graticle, stage micrometer is always given to you, but let's take it further. Right, but let's check your knowledge, guys. A graticle and a micrometer scale can be used to measure the size of biological structures that are viewed with a microscope. Which row shows the correct location for the placement of graticle and the micrometer scale on the microscope shown? Remember what I told you last, okay, the last slide. Where do you put your graticle? Of course, where? Pause the video and try. The graticle is called eyepiece graticle. So which part is eyepiece? So it is a scale embedded on eyepiece. 1 here, 1. Stage micrometers, okay, is on the slide on the stage. So 1 and what? 3. Okay, 2 is an objective lens. Don't put it on the objective lens, put it on the stage. So the answer is B. All right, let's quickly look at it. So if you want to measure a cell, now these are cheek cell. I don't know, is that my cheek cell? Or yes, take these cheek cells. Of course, the first thing you do, right, this is the eyepiece graticle, very tiny. See, 10 divisions from here to here, 10. Okay. All right, first of all, measure the size of the cell. So if you measure the size of this cell now, from 40 to 50 to 60, 40 to 50 is 10 division, 50 to what? 60 is under 10, that's 20. So that means get the size of the cell in eyepiece graticle unit. Remember, that's calibration. So it is 20. If you check, this is 20. Okay, eyepiece unit. Now the eyepiece unit is not real unit, it's not micrometers. You have to convert it to real unit, and that's calibration. Calibration is conversion. All right, So you now remove, see what we do now, remove this, the slide carrying our specimen, and then replace it with the slide carrying the eyepiece graticle. So this stage micrometers. So you see, and clash the two scales. The slide that was here was carrying our specimen. We just use the eyepiece graticle to measure, and we got 20 divisions. We'll remove this slide and place the slide having our stage micrometer, and then clash the two scales. Find point of overlap. Can you see from here to here? Now can you see from now two of them overlap here, two of them overlap here. So you see within that overlap, if you check eyepiece from here to here is 40 divisions, because from here to here is 10, 20, 30, 40 divisions. But for this from here to here is 0.1 millimeters. Can you see? According to this is 0.1 millimeters. Now once you get that overlap, remember that in calibration we want to find the value for one eyepiece graticle. Okay, so that means that 40 eyepiece, 40, okay, EPU, according to what we have there, okay, equals 0.1 millimeters. Sorry, 0.1 millimeters. So that's all right, of stage micrometer. All right, so remember we need to convert that 0.1 millimeters, convert it to micrometers. So how do you convert millimeters to micrometers? Remember what I taught you? Multiply by what? 1000 factor. Okay, multiply by 1000, right? And then 0.1 times 1000 give you what? That's 100 what? Yeah, 100 micrometers. So we have converted that, right? Yeah, sorry for, okay, so I can remove it. I say 40 eyepiece units. Come with me, let's play this music. 40 eyepiece units, guys. Okay, is 100 micrometers. See, that's what this means, because that point of overlap tells us that 40, remember, don't forget this, point of overlap is very important. From here to here is 40. Now they overlap here and overlap here. For eyepiece from here to here is 40. For our stage micrometer from here to here is 0.1 or 0.1. Convert it to micrometers, because micrometers is the basic unit we use for calculation. Okay, all right, so don't forget, now, so remove this 0.1, bring 100 micrometers. Okay, so our calibration now is if 40 eyepiece is 100 micrometers. All right, so what will be the value of one eyepiece division, which is what we're interested in? That's what calibration. So one eyepiece is what, guys? Okay, it will be equal to, it's 100 divided by 40, right? So you multiply by 1 and divide by 40. Okay, cross and multiply. It's a math scale. So by the time you divide by 100 divided by 40 will give you 2.5. Okay, 2.5. So that means one eyepiece graticle unit is 2.5 micrometers, guys. 2.5 micrometers. That's what it is. Okay, so this is this. So I'll bring it here. So what will be the value for 20? Multiply 20 by 2.5. So 20 multiplied by 2.5, 20 eyepiece graticle. We want to calibrate it now. So we know the value for one eyepiece graticle, okay, which is 2.5. So 2.5 times 20. So that means that the actual size of that cell is 50 micrometers in real conventional unit, guys. Right, so we have measured the cell live and direct. This is micrometers, please. So I hope you understood. You can replay this video, but let's check our knowledge. Let's go, guys. Look at this example here. Okay, calculation example. The diagram shows a stage micrometer scale on which small divisions are 0. Now stage micrometer the small divisions, one small division, right, is 0.1 millimeters. They have told us, all right. Now the thick one is eyepiece. This is no, the thick one is stage micrometer, right? This tiny ones are eyepiece. Okay. They say the stage micrometer scale replaced by a slide of plant cells, replace it by specimen. Then I said, what is the width of chloroplast? Okay. So guys, see what we did here. The first thing you do is that find point of overlap. Can you see the overlap here? 4 and 50. Can you see? So that means from 10 and 50, from 10 to 50, how many divisions? That's 40. Abi, that's 40. From here to here, 10, 20, 30, 40. So 40 eyepiece, okay, equals 1 sm. That's what we're trying to say here. 40 eyepiece equals 1 sm, and that 1 sm is what? 0.1 millimeters. So you see, 0.1 millimeters is equal to 40 divisions of eyepiece. So if you want to find one division, you now divide 0.1 by 40, and then what do we have here? 0.0225. But you have to always convert to micrometers. As a matter of fact, start by converting. You can convert this to micrometers to have 100. All right, but the way we did it here, okay, so you divide that 40 by 0.1 to give this, convert it to micrometers. All right, multiply by 1000 gives 2.5 micrometers. Okay, that is the value of one eyepiece. Remember, the first one we did, find point of overlap and check the numbers of eyepiece that overlap with number of stage micrometer. Stage micrometer is from here just 1, and 1 is 0.1 millimeters. Okay, equals eyepiece is 40. Okay, from here to here is 40 divisions, right? So 1 is what? 0.1 divided by 40. Convert to micrometers by multiply by 1000. So now if you go to, because they say what is the width of chloroplast? This is chloroplast, this is that guy. If you check a width, is about 4 divisions. If you check it, that's 4 tiny divisions, right? 4 tiny divisions. So if one eyepiece division is 2.5, 4 will be what? That 2.5 times 4, that gives us 10. So the size of these 10 micrometers. Okay, let's solve one more example, guys. So let's do this. Pause the video and try it. Remember, the first thing you do, so the diagram shows a stage micrometer with divisions 0.1 whatever millimeters apart, viewed, right, eyepiece containing a reticle. The area of the field of view of the microscope can be calculated using, this is the area of the field of view. A student calculated the area of the field of view of the microscope using the information and the value, that's pi. Which answer has been rounded correctly to arbitrary number of significant figures? So what we're looking for is area, guys. Remember, you have to calibrate first. Remember, find point of overlap. From here to here is also 40 for eyepiece. 40 eyepiece is the same thing as 1 sm. Okay, so but watch this. Remember that we have been given that area is what? Pi r squared. Now what is r? Radius. The radius of this circle is from the circumference to the center. Okay, that is from here to here, and from here to here, from 0 to this is 50 divisions, right? Remember that, guys, we have settled that 40, watch this, we settled it here, that based on overlap, from here to is 1 sm equals to 40 eyepiece. Okay, so 1 sm, or can say 40 eyepiece, all right, just like we did the other one. Eyepiece units is equal to what? Is equal to what? 0.1 millimeters. Okay, convert it to micrometers. Okay, by multiplying by 1000, which is something as 100 micrometers. Because if you multiply 0.1 millimeters by 1000, we want to convert it to micrometers. So multiply by 1000, we have 100 micrometers. 100 micrometers. Okay, 100 micrometers, right? So that means 40 eyepiece is the same thing as 100 micrometers. Is that okay? Now we're looking for radius. Radius is 50. So you see the radius here is from the circumference. Look at it. Circumference to the center. And circumference to the center is 50 eyepiece vertical. So if 40 eyepiece vertical is 100, 50 will give us what? Do you get? So just go straight that way. So that means 50 will give us what? 50 eyepiece unit, which you want to find out radius. If you like, you can get for one eyepiece vertical, or want to go straight, right? So 50 will be 50 times 100, right? Yeah, cross and multiply. So 50 times 100 over what? Over 40. So let's work it out. All right, this cancels this. 2 here is 2. 2 there is what? 5. Okay, so 2 here is 1. 2 into, let's just multiply it, we're going to get 25. So 25 times 5, right? Just use your calculator. Now 25 times 5 will give you 125 micrometers. That is the radius. Okay, we have converted that radius, which is 50 divisions, to real value. Now that's our radius, guys. And don't forget that area is okay, pi times radius square. So that means the area is equal to what? Come help me, guys. Pi is what? This is pi 3.142. 3.142. Very important. Okay, now this is point, it's not 1. This is 3 point, this is 1. 3.142 times radius square times radius square. Radius is 125. 125 squared. So that's 125 times 125, guys. All right, so get your calculator and help me out here, guys. You've got to do something. This is division of labor. Do not abandon your man. I'm your man, Shedro. So guys, so if I say now 125 times 125, okay, times 3.142, that gives me what? Basically it gives me, if you punch your calculator, it's going to give you 49. Okay, 49, all right, thousand, whatever you call it. Okay, is it 49 million or whatever? 49,000, 4909, okay, 3.75. okay, that's what it gives me. Now the question says which answer has been rounded correctly to an appropriate number. So if you, now let's go. So if you get it, this is going to be 1 to standard form, 2, 3, okay, 4. So this is going to be 4.9. okay, 4.9. Now they said to significant figure. 0 is not significant figure. So instead of writing, if it can be, you see 4.909, right? But this 0 is not a significant figure, so round it off to 4. Oh, sorry, let me write it very well. 4 point what? 4.91. That's significant figure. All right, micrometer squared. Is that okay? Micrometer squared. That's micrometer. That's U. So that is D. D is the answer, guys. Let's go. So we have talked about all of this. All right, stay with me. So you see resolution, ability to distinguish between separate forms of information about limit of resolution, wavelength, radiation used to view the electron, have extremely short wavelength. Okay, we have done all of this. Now, guys, I want to mention this. Remember, this is an electron micrograph. They say an electron micrograph. Now, electron micrograph there are 2 types of electron micrograph. Quickly, I want to show you something. There are 2 types of electron micrograph. We have what is called transmission electron. Okay, TEM. Okay, and when I finish, I'll ask which one is that. And we have scanning electron micros. Okay, That's scanning. Scanning is SCANNIG. Okay, scanning electron microscope. Okay, so this term gives us image in 2 dimension. Okay, 2D. The image is 2D. The image in SEM is 3 dimension. Okay, length, width, and height. But this STEM has high resolution. It shows us ultra structures. Okay, higher resolution, right? This gives us low resolution. Okay, so TEM shows tiny section of the specimen. Okay, this shows us the SEM shows us the surface contours. So looking at this structure, which one is it? The first question I ask you is it 3 dimension or 2 dimension image? Of course it's 2 dimensions, it's not 3 dimensions. So that's what we call transmission electron micrograph. Okay, and of course it has high resolution. You can see mitochondrion, central, right? So we can see the ultra structures of cells there. Okay, another check says the electron micrograph shows some of the structures. Some cell structures are listed. Which cell structures can be seen in the electron micrograph? Pause the video and try it. Which one can be seen? Okay, let's check. Nucleolus, where this is nucleolus, is the darkly stained part of the nucleus, right? It can be seen. So 1, A and B has 1, so C and D eliminated. Chloroplast cannot be seen here, if you don't, okay? And then microtubules cannot be seen. Ribosome, all these tiny spots. Okay, now when I say microtubules, they are not visible here. I mean, they are not present. Okay, yeah, chloroplast is not present. It's not that it cannot be seen by electron microscope. So, but you can see tiny dark spots. Okay, that's ribosome. Mitochondria can be seen. This is mitochondria, right? See it. Plasmodious matter is a link between a cell and another. This is a plant cell. They have plasmodious matter. You can see that link out there. So we see 1, 4, 5, 6. 1, 4, 5. So the answer is Bravo B. Okay, so see, just want to define organelles. So what are organelles? What are organelles? Try to define organelle. Okay. Yeah. So organelles functionally, structurally distinct part of a cell, usually bound, membrane bound, main features of animal plant cells view under school simple light microscope. So you can see. So what is their similarity? Okay, they have cytoplasm, they have nucleus, cell membrane. What is their differences? Okay, plant cells have chloroplast, this doesn't have. Plant cells have large central vacuole, this has small temporal vacuole. Plant cells have cell wall, animal cells doesn't. Okay, but animal cell also has central, is not here, but plant cell has plasmodious matter, this doesn't have. Okay, so let's go into cell fractionation quickly. Now, cell fractionation. If I give you an apple fruit and I ask you get me the mitochondrion, can you do that in the lab? I give you a liver from a cow that was butchered. Say get me the nucleus of these liver cells. Can do that. Now that's how we study, right, cell organelles. So separating out cell component based on their size and weight, that's called cell fractionation. Okay, 2 major techniques. The first one is called it homogenization or blending or transmigration. So what do? Get the liver or apple, put it in a blender and then grind it. Okay, or put it in a mortar and use sand to grind it. The reason for this, is to break the cell to release the cell content. Okay, so when you are done with homogenization, guys, watch this. You're going to have a homogenate. Homogenate is a solution. So you're going to have a homogenate. Okay, homo, H-O-M-O, homogenate. So homogenate simply means solution. You get, now this homogeneous has a lot of cell structure there. Okay, so this homogeneous should be stored in ice cold. Don't forget it. Ice cold. Okay, ice cold, stored in ice cold isotonic buffer. Yeah, store it there. Isotonic buffer. Ice cold isotonic buffer. Okay, so you put in a buffer. Buffer. Why, guys? Ice cold isotonic buffer. So ice cold isotonic buffer. You can see ice cold isotonic buffer. Right? So ice cold, you lower the temperature so that the enzymes, digestive enzymes from lysosome will be inactivated. If not, they will digest your organelles. Okay, isotonic, right, maintains the shape of the organelle if the homogenate is hypotonic. All right, hypotonic, that means the organelles will take up water and thus if it is hypertonic it will, so if it is high, the organelles will lose water and shrink. So buffer maintains pH, all right, so that essential enzymes, all right, do not denature. Okay, so you put this homogenate in a centrifuge test tube and then you put it in a centrifuge and carry out the next stage, which is ultracentrifugation. So remember, the first one is blending or homogenization. Now, after you centrifuge, spin it at very low speed. Guys, remember, the secret is that the heavier the organelle, the lower the speed you need to get it out. The heaviest organelle comes out. Okay, so after spinning with low speed to get the heaviest organelle, you're going to have thick sediment here. Okay, the thick, this is the heaviest organelle settle here. We call it pellet. The pellet settles at the base of the tube. Okay, that is the first pellet you're getting is the heaviest structure, right? Pellet is thick sediment at the bottom, and then we have a solution, okay, above the pellet, a bit transparent or thereabouts. Okay, this solution here is called supernatant. So supernatant contains other structures yet to be separated. They are all there. Supernatant solution right above the pellet. So guys, what do you do with this supernatant guy? Right, what do you do? Supernatant, can you see it? SPUERNATN. So the supernatant is drained into another, okay, test tube, and then spawn. It is drained and then spawn at a higher speed to separate the next heaviest organelle. Okay, so that's going to be our pellet 2. I'll call it P2. All right, and then the supernatant of this is drained in the next tube and spawn again in a higher speed again to get our pellets. Remember, the pellets are the heavier organelles separating out. That's how we're getting our organelle. This is P2. Okay, that is P3 now. All right, this continues until the last organelle is separated out. How do you know the organelles that are separated out? Now, there's an acronym I use. I designed it. It's called C, nucleomylar. Guys, remember that some of the materials we're using here, okay, came from your textbook, right? That's Cambridge textbook by Mary Jones, Richard Fosbury, Dennis Taylor, and Jennifer Gregory, and they were using some part of, okay, your Z notes. Okay, so now but this one I coined it out. Okay, nucleomylar. So this shows us structures that ask a lot, and you know, they arrange in their size, okay, or weight. Okay, weight. So this C, if we are dealing with plant, C is, okay, yeah, plant, like apple. Okay, the first one that we're dealing with plant, like your apple. I gave you where is my apple fruit? Now this is a plant apple. Okay, I say you should help me get me the mitochondria of this apple. So if it is yeah, sorry, if it is yeah, a plant, of course C works. C represents cellulose, right? Cellulose. That's cellulose. C is cellulose. Okay. Yeah, cellulose. So cellulose is the heaviest structure that will come out. Nuc, nu represents nucleus. The second heaviest, nucleus, nucleus, right? Nucleus, nucleus, right? And then chlo is chloroplast. The third one is chloroplast. Chloroplast shows up. You know the spelling of chloroplast? Okay. Yeah. All right. Let me stop at chloro. Now this is Mi is mitochondria. Okay. Mitochondria. Is that okay? Mitochondria. Now Li is lysosome. L is lysosome, right? Lysosome that carries digestive enzyme. Lysosome. E is endoplasmic reticulum, guys. Okay. Endoplasmic reticulum, ER, and R is ribosome. So ribosome is the smallest, okay, or the lightest, ribosome. Okay, so guys, let's play this game. Watch this. So you see, so if I'm doing with plants, so this is going to be pellet 1 is cellulose. That's the first one to come out. I'll spin at higher after separating it. Pellet 2 is going to be my nucleus. So get the nucleus. Pellet 3 is chloroplast. Okay, if it's a plant like apple. Pellet 4 is mitochondria, which is okay, responsible for aerobic respiration. So pellet 5 is lysosome, okay, that digests organelles. Pellet 6 is endoplasmic reticulum, that's P6. Okay, and then pellet 7 is ribosome, P7. Okay, don't forget that. All right, if it's a plant, okay, but if it is animal, if it's an animal like the liver, if it's an animal tissue or animal organ I gave you, animal, right? There's no cellulose there, animal. So that means this becomes P1, nucleus. Becomes P1, no chloroplast, mitochondria becomes P2, okay? Yeah, nucleomyla. And lysosome becomes P3, okay? See, it's changed. Endoplasmic becomes P4, and ribosome, the smallest, becomes P5. Don't forget it, guys. Okay, so this is heaviest, right? This is lightest, lowest. Okay, so watch the video again. Watch. Let's play game now. Knowledge check. An experiment was carried out to separate the cell structure in an animal cell. The cell were broken, the extract was filtered and put into centrifuge tube. So this is blending, okay, homogenization. Tube, ultracentration. This tube was then spun so that the heaviest cell structure sank to the bottom first, forming pellet 1 as shown. This pellet 1, remember, is an animal cell. So pellet 1 is going to be what? Nucleus, of course. But pause the video and try it first. Pause the video. The liquid above pellet 1 was poured into a clean centrifuge tube and spun again at a higher speed to separate the next heavier cell structure. This cell structure sank to the bottom, forming pellet 2. This procedure was repeated twice to obtain pellet 3. Pellet 4 each contain a single structure. What is the function of cell structure extracted in pellet 1? I just told you. This is animal cell. Pellet 1, check the previous slide. Pellet 1 in animal cell is nucleus. What is the role of nucleus? Digestion of old organelles? No. This is lysosomal. Production of ATP. This is mitochondrion. Production of mRNA. Yes, that's where transcription occurs. mRNA is from the nucleus. The synthesis of protein, that's ribosome. Okay, which is the last pellet. So the answer is C. C. All right, now let's check 13b. A scientist carried out an experiment. Now I want you to try this. Pause the video and try this, okay? A scientist carried out an experiment to separate the organelle in an animal cell by mass. The scientists mixed the cells with a buffer solution which had the same water potential as the cells, that's isotonic. The cells were broken open with a blender to release the organelles. The extracted mixture was filtered and then spun in a centrifuge at a higher speed to separate the heaviest type of organelles. This sank to the bottom, forming solid pellet. You can see that. The liquid above the pellet one was, now remember we're doing animal again, was poured into a clean centrifuge tube and spun in the centrifuge at a higher speed to separate the next heaviest type of organelle. These organelles sank to the bottom, forming solid pellet 2. This procedure was repeated twice more to obtain pellet 3 and pellet 4, each contain a single type of organelle. What is the main function of the type of organelle extracted in pellet 2? Remember, pause this video and try it. Animal cell, pellet 1 is nucleus, pellet 2 is mitochondria. Mitochondria, what's the function of mitochondria? Digestion of old organelles? No. Production of ATP? Yep. Yes, that's it. All right, now we are coming to the end of this show. So this is the last one. We're looking at plant diagram. In microscope, you must draw the plant diagram. Okay, so a plant diagram simply means outline of tissues. Okay, shows layers of tissue. It shows only layers of tissues. Each layer of tissues is represented with two lines. That lines must be thin, continuous, unbroken. Okay, no tails, no cells, no organelles are drawn. Okay. So for example, I'm going to show you an example. Now, if I have, for example, watch this. I have these cells. Remember, it's outline of tissues. These cells, they are like, so they form a tissue, right? Right. Now, I have another different kind of tissue, right? These ones are tall. Okay, see that. Tall cells. So that tells you they're quite different from the first one. And I have another one here. Okay. I have another one here. Okay. Okay, and I have another one here. Okay. All right. So if you check, if I'm to draw the plant diagram of this, okay. Now this tissue here, I'll represent this tissue using two lines. Okay. Yeah. So this is just two lines. Okay, and then these proportionally. Now these two lines represent this tissue. Remember, shows layers of tissue. Each layer of tissue is represented with two lines. Okay, so the next one I use two lines, so I just add another line to that. So layer 2 is going to be like this. Okay. Okay, remember this arrow. So layer 3 proportionally, layer 4 something like this. So see, what I just did now is that this is the plant diagram of this. So plant diagram does not show cells, it shows layers of cells, outline of tissues. This is a tissue, two lines. This is another tissue, two lines. Another tissue, two lines. Okay, now this is the outline of a leaf. I didn't show it, but sometimes you find, of course you should find it. You find the vascular bundle here. And this part, that part is spongy mesophyll side. Okay, spongy, right? So you find the vascular bundle there. Okay, so the first there is, of course, watch this. So this is epidermis, upper epidermis. This is palisade mesophyll. This is sponge mesophyll. Now in sponge you find the vascular bundle. This first part is xylem is the first. Okay, the middle is cambium, the inner is phloem. But if I decide to draw for on that of stem. Okay, so I'm gonna have, of course, we have epidermis. Okay, sorry. Then I have cortex. The next layer is cortex. Okay, so we have video on plant diagram. Okay, advanced practical skills. Okay, check it here. So this is epidermis, right? Now, but because this is stem, so vascular bundles are not at the center. This is vascular bundle. Okay, so this is a vascular bundle. They're not at the center. Sorry. Okay. Yeah, vascular bundle here, right? So yeah, so you can see that. Yeah. Okay. So now that's for stem. If I want to do um for roots, because they are similar, that's why I'm going to do that. You see, these are showing, this is just showing the layers of tissues. It's not showing me cells. So that's why, sorry, call it plant diagram. The lines shouldn't touch, All right, have fantastic drawing too. Yeah, where are you? Oh, okay, let's try it again. Yeah. Okay, let me just go this way, showing that it is root. Okay. Yeah. So this is epidermis, right? Epidermis. The root hairs. You can see the root hairs. Okay. Root hairs. Yeah, root hairs. Right. So we have. You can see that. Cortex again. Sorry, the lines should be joined. Okay. Now this is epidermis, endodermis. Oh. It's too big. Let me delete this and do something better. So thank you for staying true, but guys, we need to master this. Okay, I know I've taken your time, okay, but I want you to become the best. So this is endodermis. Okay, of course, the vascular bundles are in the middle, xylem is in the middle here, right? Phlegm. Yeah, so just something in the middle. So you can see that. Yeah. So just follow me now. Watch this. This is leaf, stem, root. Oh, this is upper epidermis, palisade mesophyll, spongy. This is vascular bundle. Now, when it comes to vascular bundle, the outermost part is the xylem cambium. Now, phloem is inside, xylem is out, phloem is inside, xylem is out. Now, of course, this is lower epidermis. Now, if you come to stem and root have something in common. They both have epidermis, right? They both have cortex. Here is cortex, there is cortex, right? They both have endodermis, here, endodermis, right? But you see, when it comes to this, the vascular bundles are not at the center. So the one inside here is xylem. Xylem is out here. Xylem is inside in stem. Okay, phloem is outside. In between is cambium. This is called pit. Okay, when you come here, xylem is at the center, inside. Xylem is inside in stem, inside in the root. Okay, outside is phloem. Outside is phloem. All right, let's bring this to a close. Watch this. The diagram shows the transverse section through parts of the plant. Which correct? Now, which one contains lignin, is xylem, transverse organic solid is phloem. Remember, this is a leaf, this is a stem, this is roots, this is the root hairs. Okay, in the leaf, xylem is outer. So one is xylem. Okay, so the ones that have one in the leaf, xylem, choose it. In the stem, xylem is inside. Four is there as lignin. In the root, xylem is also inside. 6. Okay, 1, 4, 6 is xylem. Phlegm is in the leaf is inner 2. Okay, phlegm is 2, and in the stem and in the root is outer. So 3 is phlegm, 5 is phlegm. So 2, 3, 5. Xylem is 146. Okay, thank you for staying true. Okay, so please kindly watch the next part and like, subscribe, turn on the notification video. Okay, see you next class. Bye.